For our project, we picked the enzyme trypsin.
We're going to start by going over basic information about enzymes in general and our enzyme of choice, and then move on to the function of trypsin. Then we're going to lead with some bioinformatics and end with research.
Wilhelm Kuhn discovered the enzyme trypsin in 1876 when studying pancreatic cells. Trypsin's job is to aid in digestion. Trypsin was found in the pancreas because that is where the other digestive enzymes are created. Trypsin is a protease which is found in the small intestine of humans and animals.
Now we're going to go ahead and go into our general enzyme overview. Enzymes are proteins that speed up the rate of reaction in the conversion of substrate to product. In speeding up the rate of reaction, the enzyme decreases the activation energy needed to carry out the reaction. The process works by the enzyme binding to the substrate at the active site. The active site normally follows the induced fit model, which shows the enzyme conforming to the shape of the substrate. Once the substrate is bound, it is converted to product and is released. Enzymes can be regulated, changing the amount of products that are produced. This can be done by either inhibitors, which will decrease the activity of the enzyme, or activators, which will increase the activity of the enzyme. One type of inhibitor is a competitive inhibitor. Competitive inhibitors work by binding to the active site of enzymes so that the substrate is unable to bind to the enzyme, which will ultimately decrease the enzyme's activity. This idea will be discussed later in the presentation. Now we're going to focus more on our enzyme of choice, trypsin. Trypsin is located primarily in the small intestine, but is produced by the pancreas with the help of pancreatic acinar cells. Enzymes can exist in an active state called zymogens. In the case of trypsin, this inactive form is trypsinogen. Trypsinogen is mainly found in the pancreas and is activated in the small intestine by enteropeptidase. Enteropeptidases cleaves around 7 to 10 amino acids of the N-terminus group of the trypsinogen, which induces a conformational change which will activate the enzyme into trypsin. As previously discussed, enzymes are subject to inhibitors that decrease the activity. This holds true for trypsin, which has inhibitors such as pancreatic secretory trypsin inhibitor, also known as PSTI. This is a competitive inhibitor that binds to the active site of trypsin and decreases its activity.
There are known splice variants of trypsin. One example is from an experiment done on trypsin and chymotrypsin. The experiment utilized an analog of trypsin inhibitor, which was made up of two peptide chains for the N and C terminus, which was brought together by a disulfide bridge. The trypsin and chymotrypsin were incubated with the analog of trypsin inhibitor. It was found that the single peptide bond between the lys and SIR residue at the C terminus underwent enzyme catalyzed degradation and left room for a new peptide bond to form. This made sure that the two sides of the peptide bond stayed together. This form of splicing is similar to other proteasomes. An example of a proteasome is serine, which is very similar makeup to trypsin. So now we're going to talk a little about the function of trypsin. Trypsin is a serine protease, meaning it cuts proteins. It is secreted by the pancreas and plays a part in digesting food. Trypsin is found in all animals, including insects, fish, and mammals. The proteins humans eat are chains of amino acids. These amino acids have a carboxyl terminal and an amino terminal at each end, which allows them to form links of amino acids. Once a person eats the protein goes into the stomach. Once the food is in the stomach releases pepsinogen and HCl. The HCl will trigger the conversion of pepsinogen to pepsin. Pepsin is what hydrolyzes peptide bonds, meaning it breaks the bonds into amino acids. This breaking of the bonds breaks down proteins into large polypeptides. These peptides are then taken to the small intestine, which is where trypsin comes in. So now we're going to talk a little about the mechanism of trypsin. Now food, which has been broken down into large polypeptides, are in the first part of the small intestine, the duodenum. Once here, the pancreas secretes trypsinogen, a zymogen, which as mentioned earlier is a fancy way of saying it is a precursor to the active enzyme form. So it will become trypsin, but it's currently inactive. Trypsinogen goes through the pancreatic duct and into the duodenum. The duodenum has mucosa that contains enteropeptidases, which is what cleaves the peptide bond of the trypsinogen after lysine, which is the 15th residue. Because of this, the N-terminal residue is kicked off and a new one is made. The removal of the amino acids at the N-terminal induces a change in trypsinogen and the trypsin is formed. Once we have trypsin, a substrate can start binding to it. When a substrate is bound to the active site of trypsin with the help of water and the amino acid serine, trypsin catalyzes the hydrolysis of peptide bonds, which creates peptides and amino acids. Trypsin is interesting because at the bottom of the S1 pocket, it has a negatively charged side chain from aspartate. This gives it the ability to only cleave at the carboxyl end of the amino acids, which has a positive end from an arginine and a leucine. The positive charges of the R groups on these amino acids interacts with the negative charge of aspartate. When the positive and negative charge come together, they stabilize each other. This is why it can only cleave at the carboxyl end of lysine and arginine. The presence of trypsin in the small intestine stimulates more trypsin to be formed. Also, it helps convert chymotrypsinogen, a zymogen, to chymotrypsin, and pro-carboxypeptidase, a zymogen, to carboxypeptidase. Chymotrypsin and carboxypeptidase are similar to trypsin in aiding with digestion. So trypsin is a triple threat because including itself, it stimulates the activation of 3 digestive enzymes. To further study trypsin, we use the bioinformatics website Swiss Model. Swiss Model is a bioinformatics website used for protein modeling. Before running the analysis, a target sequence or the UniProt of the protein is required, which we got from BLAST. To perform its function of protein modeling, Swiss Model goes through 4 steps. First, it identifies the structure of the template. Then the target sequence and template structure have to be aligned. The model is then built, and finally, the model is checked for quality control. Swiss Model operates by utilizing protein sequencing, structured databases, and specialized software that is continually updated to ensure accurate results. The protein model of trypsin is above. In the website, when you put your cursor over the model, it will tell you what amino acid you are at and the location. Protein modeling is important because the shape of an enzyme has a direct effect on catalysis. Protein modeling is especially useful in making new medications. Knowing the structure of a protein can help in developing enzymes for drugs. This isn't just to make the drug work better, but one common use of protein modeling is to figure out the structure of enzymes to help make a new enzyme that aids in the efficiency of making drugs, which in turn makes medications cheaper and more widely available.
Now we're going to use Swiss Model to focus more on the structure of trypsin. From Swiss, we see that trypsin has a globular structure. However, it is only composed of one polypeptide with 223 residues. In the structure, there are a number of cysteine bonds present, which allow the molecule to participate in disulfide bonds between itself and other molecules. Trypsin's active site is composed of serine, histidine, and aspartate. All of these residues are polar with a slightly negative charge, which will likely attract slightly positive residues such as lysine or arginine to its active site. We're now going to go ahead and jump into the research that we're presenting. This research focused on pancreatitis. Pancreatitis is the inflammation of the pancreas. This inflammation is caused by the early activation of trypsin in the pancreas. If you remember from earlier in the presentation, we discussed how the inactive form of trypsin, trypsinogen, is mainly found in the pancreas, but is converted to trypsin in the small intestine. So in this case of pancreatitis, trypsinogen is converted in the pancreas, which causes irritability in the pancreas, which leads to inflammation. This experiment is focusing on the different ways in which trypsin can cause pancreatitis. The first method that was discussed was early trypsinogen activation. The journal focuses on ways in which trypsinogen can be activated too early on and which will cause in ways such as mutation. The next method was inhibition of trypsin. Pancreatic secretory trypsin inhibitors, also known as PSTI, is a competitive trypsin inhibitor that was previously discussed in the presentation. This inhibitor is used to regulate the production of trypsin. However, in the case of mutations in this molecule, trypsin will continue to be created without stopping. This will lead to overproduction in trypsin, which will result in pancreatitis. The last method focused on trypsin receptors, specifically protease-activated receptors, also known as PAR2. This receptor presents once pancreatitis is starting to develop. When pancreatitis is sensed, PAR2 works as a negative feedback to slow down the production of trypsin. Any damage or mutation to this receptor will take away a line of defense against pancreatitis before it fully develops. In the article, the researchers tested the activation of trypsinogen to trypsin and its regulation by PSTI and the onset of pancreatitis. Now we're going to go on to the methods. This experiment was done by gene targeting and infecting the PSTI gene into knockout mice. Once the gene was injected, the researchers analyzed the phenotypes of the mice. The results. Researchers observed that the mice with PSTI gene maintained the integrity of the pancreatic acinar cells by inhibiting the production of trypsin too early on. This proved true because the mice had less pancreatitis rates than they normally did. When there was a mutation in the PSTI gene, it cannot inhibit trypsinogen, and the pancreatitis was observed. They specifically found that missense mutation on the PSTI gene resulted in a conformational change within the molecule, which prevented it from binding to trypsin. The results also introduced the idea of autophagy, which sensed trypsin for degradation. It determined that autophagy could result from uncontrolled trypsin activity, or activation of trypsinogen may occur once autophagy is initiated. Ultimately, the study proved that the disruption in trypsin and its inhibitors could result in pancreatitis. Here are our references, and thank you for watching.